Okay, so thanks a lot for the kind invitation to talk here. So it's a pleasure to be back in Korea. This is my third time here. And I'm particularly pleased to talk here because the main research direction of my lab now has been shaped very directly by interactions here at IP Korea, in particular with Su Jin Jong and also Spencer, our late colleague Spencer Shorter. So I will tell you about the work of my lab in developing deep learning approaches to help find novel antibiotics, but this is very much work in collaboration with multiple teams, in particular Ivo Boneka, who is here, and others. So since time is limited, I will probably skip the introduction about why we need novel antibiotics. Ivo already stressed this. The point that I would like to highlight is that we need novel antibiotics that have new modes of actions in order to escape the current resistance mechanisms. And unfortunately, the antibiotic discovery pipeline has dried out with very few exceptions. So we really need not only antibiotics with novel modes of actions, but it's more desirable to actually have a process, a pipeline that can deliver new antibiotics with new modes of actions as resistances are going to continue to evolve and as we may face a new pandemic, which may be bacterial this time. So the current mainstream approaches to antibiotic drug discovery have been mentioned already. They led to some successes, but also have limitations. So the target-based drug discovery, requires a target to begin with. That's an important limitation. And then, screening for drugs in vitro that will lead to strong interactions doesn't mean that this drug can be then effectively used in bacteria. So the alternative approach of phenotypic drug discovery, so typically using growth assays where you just screen for drugs that or compounds that are going to, kill cells or limit their growth, has been arguably more successful, but doesn't give us insights into the mechanisms of action, which is a key limiting factor, a key step in the drug discovery pipeline. Also, the chemical space, it is gigantic, 10 to the 30 or more compounds has been explored. Only a small, tiny fraction of it has been explored. So, my lab tries to contribute to this project that aims to overcome some of these limitations and by relying on phenotypic approaches to characterize bacteria, but also to use these phenotypic approaches in combinations with AI methods, and AI these days means deep learning, to predict from phenotypic data, to predict modes of actions or even molecular targets of compounds, and then use this approach to explore larger fractions of the chemical space. And as I mentioned, this is work in collaboration with Ivo Boneka, but also Mark Ponstrup, a chemist in, at the HCI in Braunschweig, a specialist of antibiotic drug discovery, also with a focus on natural products. And our goal here is to design a pipeline that will be able to analyze compounds and try to determine their mechanisms of action or targets in a very hopefully rapid manner, and therefore to hopefully accelerate antibiotic drug discovery. And the core idea of this project is to rely on imaging, but also Ivo is setting up metabolomic techniques. So to rely on data analyzed by deep learning to determine fingerprints of modes of action. So more specifically, our strategy is highlighted in this slide, and it very much is based on mutants. So our goal is to characterize phenotypes of bacterial mutants, ideally of all genes in the genome, which we're going to knock out or knock down one by one, and then to use imaging to characterize the phenotypes of these mutations, and then use deep learning to transform the data into a high-dimensional space that will allow us to extract these digital fingerprints, ideally one per gene, but more realistically, we expect to get fingerprints that will reflect groups of genes, part of the same pathway that will lead to the same phenotype. And once we have mapped out this kind of phenotypic landscape, we will use it to explore chemical libraries, so to look at wild-type cells exposed to different antibiotic drugs, and then use the same approaches, typically imaging and deep learning, to then project the data in this same phenotypic space, a so-called latent space, and then, if a specific drug, like say compound X, leads to a phenotype, so a latent, an embedding here, that is similar to mutants of, let's say here, gene cluster C, then the prediction would be that compound X targets a bacterial protein that is encoded by one of the genes in this cluster. So this would be a way of predicting the target or a few potential targets for each compound. Of course, this is based on the hypothesis that chemical perturbations and genetic perturbations can be matched, right? And yes, I just wanted to highlight that this core idea of the project has emerged from discussions we've had here many years ago, together with Sujin Zhang and Spencer Shorter. So we are at the early stages of this project, but what I'm going to show is that we have some first data that correspond to some of the first milestones here of this project. So the first question we asked is whether the images actually contain these fingerprints that we're looking for. Do they contain information that is specific to particular modes of actions? Because if this is not true, then the entire project would fail. Now we started, and a secondary question is that if the answer is yes, then what kind of images do we need to characterize these specific fingerprints? And what kind of microscopes do we need for this, right? And there's been prior work along these lines, so we are not the first to think along these lines. So the lab of Joe Pogliano many years ago has shown that with fluorescent microscopy in combination with so-called handcrafted features, so like descriptors of the bacterial morphology, such as the length, the mean fluorescence intensity, and so on, they were able to cluster drugs by phenotypic similarity in a way that made sense. And then there was follow-up work from Roche that kind of extended this approach, again, relying on fluorescence images and handcrafted features. So we decided to look at this in the context of AI. So instead of trying to extract morphological characteristics, we are betting on deep learning, so these artificial neural networks, because they have been shown to be able to extract very subtle data from images in a way that the traditional techniques are not able to. So in practice, we started by focusing on about 20 reference antibiotics. So this is a proof of principle where we're asking, can we extract these fingerprints in a way that is specific to modes of actions? And so the lab of Ivo and specifically Kelvin Ko, who is here, obtained images using a high-throughput microscope, also with the help of Nassim Matal, who is also here. That images shown here, so in bright field on the left and on the right with fluorescent markers of the DNA and the membrane for these 20 antibiotics at concentrations of IC50 down to 1/8 of the IC50. And then So, you know, in these few examples here, you can see kind of dramatic changes in the morphology due to the effect of the drug. So you would not need any deep learning to analyze this. But as the number of conditions increases, here I'm showing only 12 out of the 22 drugs, it will become much harder to actually distinguish them. So what then my former PhD student, now postdoc Daniel Krenzel, did is that he developed convolutional neural networks pre-trained on generic computer vision data, but retrained here in a supervised way on the images of bacteria. And in this case, he focuses only on the bright field channels, so ignoring the fluorescent channels. And the task here in the supervised learning approach was to recover the identity of the drug and its concentration from the images alone. So this is a classification task with 88 different classes. And importantly, he trains the model on 4 biological replicates and then tests the performance of the model on a 5th independent biological replicate. And the results can be visualized in the form of these confusion matrices, which show you essentially the true label, because obviously we know with what antibiotics we have used and what concentration for in each well of these multi-well plates, and then the comparison with the prediction of the algorithm. And if this was perfect, you would get values of 0 outside of the diagonal. And what we get is something like this. So relatively good, but not perfect classification accuracy of between 75 and 90%, which means that the model is able to recognize some features in the images, some fingerprints that are specific to the drugs, but also with some levels of confusions that we are not happy with. So we would like to address essentially. So it happens that these 22 antibiotics were selected based on the most widely employed antibiotics. Most of them are cell wall inhibitors, and so they fall within 8, I believe, different mechanisms of action based on the literature. And you can see here, I mean, half of them are cell wall inhibitors. Here we distinguished PBP1 from PBP2 and PBP3 inhibitors. And now if we pool the predictions of the model by the mode of actions, then we get a different confusion matrix here, which is essentially perfect, meaning that we can recognize the mode of action of the drug from these images alone. And these were bright field images, so fluorescence turns out to be not needed in this case. So this led to a number of follow-up questions. One of them is, can we actually detect a phenotype at sub-inhibitory concentrations? And essentially the answer I already gave you is yes, because the confusion matrix I showed you was at IC50 concentration, but then we explored this further, and essentially the message is that at concentrations, even at half IC50, we can still detect a phenotype for most of the drugs. Then the accuracy goes down as the concentration decreases. Nevertheless, this shows that at IC50, so at some inhibitory concentration, and this model is able to pick up a phenotype that the traditional growth assays would have missed. Okay? So another question was whether we can actually, so here, the model was trained on reference drugs, and we use it to recognize the effect of one of these reference drugs, which is a bit of an academic exercise, but not really useful in practice, because you want to analyze new drugs, right? So we tested what happens if we can train the model on compounds but leaving out one of them entirely, and then testing the model on that left out compound. Is it able to correctly assign the mode of action of this new compound? And essentially the answer is yes, with the exception of the PBP1 inhibitors. But we looked at them more carefully. I mean, especially Kelvin and Ivo did Boston competition assays, which revealed that actually the annotations of the modes of actions for these 3 inhibitors were probably incorrect, at least for our E. coli strain. So it shows that the model is able also to kind of revisit annotations in the literature to some extent. So another follow-up question is what happens if we leave out a mode of action entirely? And can we detect if a drug has a novel mode of action? And again here, I skipped the details. The answer is yes, in this case with the exception of the PBP3 inhibitors, which probably have a phenotype that is too similar to the other ones for it to be distinguished without showing it to the model during training. So this was data on E. coli, but essentially Thomas Delerue, who is also here, acquired data on Klebsiella, and we get essentially the same results. So mode of action recognition from bright field images alone works very well on this species also. And so what we're suggesting is that this imaging-based approach could be complementary to the traditional growth assays. So particularly thanks to its ability to pick up subtle phenotypes, right? And it could allow us to screen compounds for that potentially have a novel mode of action compared to the reference antibiotics that already exist. And this would then lead to follow-up experiments to characterize that mode of action. But as I told you, our goal is to also accelerate this process. And so the question is, we can not only detect if a compound has a novel mode of action, but actually identify it. And for this, as I told you, we are going to rely on mutants. So the question is whether we can also extract meaningful fingerprints from the images of mutants. And so here are, so for this, this is something we started only recently, but so Felix Knote in my lab in Würzburg and also Kelvin acquired phenotyped E. coli using CRISPR-I to knock down about 30 different essential genes in E. coli. So this led to, as you can see here, different phenotypes. And if we again analyze them, in this case this was work from Richard Jom in my team in Würzburg. So here the model was trained to recognize the guide RNA that was used to knock down a protein. In this case we have 3 guide RNAs per gene, so it's an 84 class prediction problem. And again, we have a pretty strong diagonal, but the accuracy is only about 30%. Now, if we pool the guide RNAs by gene, we get accuracies of 70%, which is already significantly better. And now, if we group the genes by pathways based on gene ontologies, then we get the confusion matrix that I'm showing here, which has an accuracy of 100% recognition. So similarly to the drugs we have here, I mean, these are preliminary data, but they strongly suggest that we can also recognize the pathway in mutants. Now, the next step, and this unfortunately we have not solved yet, is to align the phenotypes of the mutants to the drugs in order to make this prediction of mode of action that I mentioned at the beginning. But this is work in progress, and we hope to get there by the end of the year. So for the sake of time, I will probably skip the summary here and just briefly mention the perspectives that we are now exploring. So together with Ivo, we're planning to scale up this effort to 7 bacterial species by phenotyping more than 100,000 different conditions, including mutants and drug treatments. We will use additional techniques beyond imaging. So beyond this low resolution bright field imaging, we will use super resolution fluorescence microscopy, but also metabolomics to analyze the molecular composition. We are also very interested in these AI-guided methods for drug design that have been mentioned before. We are working also on interpretability, which is a key limitation. And we hope also that resonates with the previous talk, that by training a model on many species, we'll be able to make predictions for novel species that, like a future potentially bacterial pathogen X. Just want to finish by thanking the people who did the work. So I highlighted mostly work from Daniel Krenzel on the analysis side based on data from Ivo's lab obtained by Kelvin and Thomas, who are here, and also the last part on work from Felix Knote analyzed by Richard Jom. And thank you for your attention.